In the last lesson, we saw how postsynaptic conductances evoked by glutamate release from presynaptic terminals were converted into postsynaptic potentials and integrated in complex ways across the dendritic arborization. We found that most unitary excitatory postsynaptic potentials, that is, EPSPs that originate from a single action potential in a single presynaptic neuron, that most unitary EPSPs are small in amplitude. They have an amplitude typically of 1 millivolt, and they last 10 to 20 milliseconds in duration. A excitatory neuron is typically at rest at around minus 70 millivolts, so something like 30 millivolts away from action potential threshold. In order for a postsynaptic neuron to contribute to further computation in the brain, it must receive those excitatory synaptic signals, depolarize beyond threshold, and fire action potentials of its own at the appropriate time. This means that many synaptic inputs must be integrated in that individual neuron. And in order to understand how that neuron integrates sensory information, we need to know where its presynaptic neurons are. We need to map the wiring diagram and the connectivity of how synaptic input arrives onto postsynaptic neurons. In this lesson, we'll begin to think a bit about how synaptic wiring diagrams can be constructed, at least in a probabilistic way, and how that might relate to sensory processing in the mammalian brain. We'll begin by considering sensory processing in the mammalian brain and its dependence upon glutamatergic synaptic signals. We'll think about vision and touch, two important senses for us humans. If we think about the sense of touch, there are mechanogated ion channels sitting in our skin inside axons of sensory neurons. Touch onto the skin causes action potential firing in the sensory neuron, and these sensory neurons release glutamate from their synaptic terminals somewhere here in the brainstem. The brainstem neurons in turn receive those excitatory glutamatergic signals and send them on to the thalamus. The somatosensory part of the thalamus deals with the sense of touch. Neurons there receive the glutamatergic signals from the brainstem neurons, and in turn they generate PSPs, action potentials, and send the sensory signals up to the neocortex, where it's thought that our conscious percept of touch is driven by the activity of the neurons in the neocortex. Again, most of that activity is driven by glutamatergic synapses. And so we have here a pathway of a brainstem synapse, a thalamic synapse, and neocortical synapses, all of which are primarily glutamatergic in nature and essential in order to convey a sensory signal from the periphery to the neocortex, where it might generate sensory percepts. Similarly, if we think about our sense of vision, this also depends heavily upon glutamatergic synapses. Within the eye, the photoreceptors that detect photons arriving from the visual field, they release glutamate onto second-order, so-called bipolar cells in the retina. These bipolar cells sense the glutamate and, in turn, release glutamate at their own synaptic terminals onto retinal ganglion cells. And it's a retinal ganglion cell that then receives its glutamatergic inputs, fires action potentials, and transfers the visual information first to the thalamus, where again a glutamatergic synapse conveys that information further to the visual cortex. And again, glutamate is a signaling molecule there. And so glutamatergic synaptic transmission is of fundamental importance of getting sensory information from the periphery to the neocortex, where it drives sensory perception. And that's true for all of our senses. In this video, I'd like to focus particularly on one sensory pathway that has some specific features that are useful for detailed analysis in terms of microcircuits and synaptic connectivity. We'll think about the mouse Whisker system, and the mouse is an attractive animal in terms of neurophysiology because of its extensive genetics and its extensive use in biological research. Mice are nocturnal animals. They live underground in tunnels, and so vision is not one of their most developed senses. Instead, one of the ways in which they gather a lot of sensory information about their immediate surroundings comes from their array of whiskers that are present on the snout of the animal. These are arranged in a highly organized manner, and using their whisker system, the animals are able to navigate in tunnels. They can see whether a hole is a sufficiently large aperture for them to enter in. They can detect the shape of objects, and their sense of texture discrimination with their whiskers rivals that of the human fingertip. So this whisker system is an immensely sensitive organ through which the mouse can learn a lot about its immediate spatial environment. Deflection of one of these whiskers activates mechanogated ion channels sitting here in the whisker follicle, and that drives action potential firing here down the trigeminal nerve. There's a first glutamatergic synapse in the brainstem, and neurons in the brainstem receive that glutamatergic signal. Postsynaptic potentials are generated. Some of the neurons in the brainstem fire action potentials, and those signals in turn are relayed to the thalamus. Again, thalamic neurons here receive the glutamatergic input. Postsynaptic potentials are generated and integrated in the thalamic neurons. Action potentials are fired in subsets of those thalamic neurons, and the information is then further relayed to the primary somatosensory cortex, where presumably whisker sensory perception takes place for the mouse. So there are 3 glutamatergic synapses that relay sensory whisker information from the periphery through to the neocortex of the mouse. And in particular, it's the primary somatosensory neocortex of the mouse that deals with whisker sensation. The organization of the primary somatosensory neocortex of the mouse is remarkable in the sense that it has obvious anatomical units that appear to be in a direct one-to-one correspondence with the layout of the whiskers on the periphery. And so the whiskers here, the hairs that emanate from individual whisker follicles, are laid out in a highly stereotypical pattern where every mouse has the same organization of the whisker layout, and indeed mice and rats have also the identical layout. And so because it's highly stereotypical, we can then label these whiskers in specific ways. And so this is called the A row, the B row, the C row, D row, E row, and equally they're considered to be arcs 1, 2, 3, and 4 in terms of the whiskers. And so this one here that's highlighted in yellow is the so-called C2 whisker. And if we now look at the neocortex across the horizontal extent of the neocortex, we're looking down on the face of the neocortex across its so-called somatotopic map, we'll see that there are corresponding units here drawn in blue that correspond to the whiskers. And again, highlighted here in yellow is one particular anatomical unit, the so-called C2 whisker representation. And these round things here are called barrels. And so this is called the C2 barrel, present in primary somatosensory cortex. And because of its obvious analog in terms of the whiskers on the periphery, one might think that sensory information relating to the C2 whisker is primarily processed in the C2 barrel column in the primary somatosensory cortex. And indeed, at the various stations in the brainstem and the thalamus, there's a nice representation also of the individual whiskers in terms of anatomically identifiable units. Now, if we look at a section of the brain where we cut through the thickness of the brain, and so before we were looking at the top of the brain and we saw that there was a nice C2 representation of the whisker, now we look inside the brain and we see that it has some depth. It's about 1 millimeter thick, the neocortex in this particular location in the mouse. And here maybe you can make out the walls of the C2 whisker representation. That's here high cell density represented here by this fluorescent stain of DAPI that stains the cell nuclei. And you can also see it here in this stain of neurons present in this area. You'll see that there are these walls here that represent the limits here of the C2 Whisker representation. And now this would be the surface of the brain up here, and here we go deep into the brain, and this is the thickness of the neocortex. It's about 1.2 millimeters thick. Now there are some obvious differences at different depths in the neocortex, and people have divided the neocortex into different layers. And so there's a layer 1, the most superficial, the outside area of the brain, that has very few cells in it. It's mainly composed of synapses, axons, dendrites, and synapses. The cells themselves begin here in layers 2 down to layer 6, and you'll see that there are different densities of cells in different layers. And here we've labeled some individual neurons that are present at these different layers, and you can see that there are some different morphologies of the cells. And so there are very small cells that are present here in this so-called layer 4 area. That's actually where the main sensory input arrives in the neocortex. And in the superficial layer, there are so-called small pyramidal cells, small excitatory neurons, and in the deeper layers, there are much larger cells with much more extensive dendrites. And it turns out that the molecular biology, the electrophysiology, and the synaptic properties of the cells in different layers are different. And so we can think of these as different cell types. All of these neurons here are glutamatergic neurons that, at their axonal outputs, release glutamate. And so they are of one cell type in terms of their neurotransmitter, but we can subdivide them into many different subtypes of excitatory neurons, and one of the ways of subdividing them is their laminar position within the neocortex that tells a lot about the synaptic connectivity, which is what we're going to look at further. Now, in order to functionally map the wiring diagram of the C2 barrel column, we first need to identify it in the living brain. It turns out that the anatomical barrel map that we've looked at so far is nice in anatomical sections, but when you look at the living brain, you simply see the surface blood vessels without an obvious indication of where different Whisker representations are. However, we can perform a simple optical imaging technique where we can shine red light on the surface of the brain. We can then stimulate the C2 Whisker. That then causes activity in a localized region of the brain, the C2 Whisker representation, and that changes in turn the optical properties of this area of the brain. There are changes in blood flow downstream of neuronal activity, and there are also swelling of different processes that affects how light enters and leaves this area of the brain. And so simply by shining red light on the surface of the brain, stimulating the Whisker, we can see changes in reflection of this red light, and it turns out when an area of the brain is activated, less light is reflected. And at the level of hundreds of microns, we can now map the location of the C2 Whisker. It causes a response in this small area here of the brain. We can then furthermore put a bit of red dye at this location inside the brain, and at later times we can cut horizontal sections of the brain and see that the red dye that we placed inside the brain is located inside the map of the Whiskers, and in particular it corresponds to the C2 barrel column inside the neocortex. And so, as one would expect, there's a nice mapping of the functional location of where C2 Whisker evokes responses and the anatomical map that matches them nicely. And we can also see that the spatial extent of this Whisker representation is really at this level of hundreds of microns. The C2 Whisker representation is maybe some 200 by 300 micrometers in extent, and as mentioned, the neocortex is about 1.2 millimeters thick at this location. So we can then do this type of experiment, inject some red dye, cut brain slices through the thickness of the cortex, and we can then begin to analyze how neurons that are located in different layers communicate with each other. And so here's a picture of a living brain slice. we injected red dye to label the C2 barrel column, you might be able to make out the so-called barrel walls here, present inside layer 4 of the neocortex. And in order to analyze synaptic connectivity, we can then simultaneously record from a number of different nerve cells. And in this particular experiment, we're recording from 6 neurons that are located here in layer 5 of the neocortex. So some depth from the surface of the brain, we go to the infragranular layers, and here we've got 6 whole cell recordings that are located on 6 different neurons. And so in each case, there's a whole cell glass electrode that's making contact with this neuron. It's a membrane potential recording, and so there's a continuity between the patch electrode and the cell membrane here, so we can record the membrane potential. In order to study synaptic connectivity, we can then initiate an action potential in one of our cells. We can simply inject current into cell 1, cause it to depolarize, fire an action potential, and then see what happens in these other neurons, whether any of them respond with postsynaptic potentials. And in this particular experiment, cell 1 released glutamate and caused postsynaptic potentials in cell 3. And that's what you see here, the action potential in cell 1 and a postsynaptic potential in cell 3. This 80 millivolts refers to the action potential trace, and the 2 millivolts refers to the postsynaptic potential. So the postsynaptic potential, as expected, is somewhere in this 1 millivolt range, lasting some tens of milliseconds, whereas the action potential is brief. It's this 1 millisecond depolarization of about 50 to 80 millivolts. So cell 1 is synaptically connected to cell 3. There's a synaptic connection. But what's not shown here is that that same action potential didn't cause any effect of the membrane potential on cells 2, 4, 5, or 6. So there's a single connection here from cell 1 to cell 3, and 4 of the others have no connection. And so cell 1 has a 20% probability of having postsynaptic partners. Only 1 out of 5 of these possible cells was synaptically connected to cell 1. And so we can think of that as a 20% probability of finding a synaptic connection. We can then, of course, go through the different cells and stimulate cell 2 and see whether any of these other cells responded. And in fact, it turns out that cell 2 drives a small postsynaptic potential in cell 1. Note now, the scale is rather different, and the time course of the PSP is also different from cell 3. There's some diversity in the properties of the postsynaptic potentials, as we've already seen. We then go through all the different cells, stimulating cell 1, 2, 3, 4, and 5 in turn, and seeing who responds postsynaptically to that action potential. And that then allows us to draw a small wiring diagram for these particular 6 neurons that we've recorded from. Here you see the dendritic structure of those cells. Here we've separated them out in different colors so you can see the structure of the different neurons. They're all excitatory pyramidal neurons, and they're connected in this particular wiring diagram pattern, where cell 2 connects to 1, 1 to 3, 3 connects to 6, but it receives an input from cell 5, 4 connects to both 5 and 6, and so there's a divergent output here from cell 1, causing postsynaptic potentials in both of these 2 partner cells that are postsynaptic to it. So we can then repeat this experiment over and over again in different layers, recording cells in cell 2, in cell 5, layer 4, and simply see what the probability of finding synaptic connections is across a population of neurons. We can't record from all the cells at the same time, so it becomes a statistical argument in the end, where through analyzing many different brains, focusing on one particular area of the brain, a nicely delimited column of the neocortex, we can get the statistical wiring diagram of how neurons in different layers, different cell types, if you like, communicate with each other at a statistical level, and that then helps us understand how sensory information is processed in this part of the brain. So here's the end result of the analysis. After recording from hundreds of pairs of neurons, we find that there's a connection probability matrix that we can construct, where we take the location of the presynaptic neuron in terms of which layer of the neocortex it's in, and we look and see how often that connects with postsynaptic neurons, again ordered according to which layer the soma is located in. And if we now start with neurons that are located in presynaptic layer 4, we'll see that they make connections onto postsynaptic cells in layer 2, 3, 4, 5A, 5B, and a little bit in layer 6. So layer 4 neurons send their axons and make synaptic connections with neurons in all other layers of the neocortex. On the other hand, if we think postsynaptically about neurons in layer 4, we'll see that they don't receive any input from layer 2, 3, 5, or 6, and they almost exclusively receive their input from other layer 4 neurons. So layer 4 is a very tight local processing area where neurons within layer 4 like to talk to each other, and they also like to send signals to other parts of the neocortical column, but they don't want to receive information from these other layers. We can also see that there's a strong diagonal component where neurons within layer 2, within layer 3, within layer 4, 5A, and within layer 5B like to talk to each other. They like to talk to their neighboring cells that are sitting right next to each other. That's one of the ways in which they make connections, simply by being close to each other. Beyond that, there's further specificity. The superficial postsynaptic neurons, they get information from presynaptic layers 4, 3, and 2. And so here you'll see that the layer 2 free neurons are receiving information from these circuits here. Probabilistically, that's where they're most likely to be getting their synaptic input from. On the other hand, if we look here in the deeper layers in layer 5, you'll see that they get synaptic inputs from almost all other layers. They get information from layer 2, 3, 4, 5, and perhaps a little bit from layer 6. And so these neurons here then are integrators. They receive information from all other parts of the cortex, and presumably they use that information to distribute some further processed signals to other parts of the brain. And so there's a high degree of specificity in the directions of which sensory information gets distributed to different cells in the neocortex, and what remains to be learned from further experiments is what the purpose of that computation is within the neocortex. If we now look at the amplitudes of the postsynaptic potentials that are found inside the C2 microcircuit, we find that there's a rather interesting distribution of unitary EPSPs. Most of the synaptic connections are very small in amplitude. They're sitting there somewhere at around 100 or 200 microvolts. So an action potential in a presynaptic neuron, on the whole, gives rise to a relatively small postsynaptic potential of the postsynaptic neuron. It's just a few hundred microvolts. Almost nothing compared to the amount of depolarization that's necessary in order to get this postsynaptic cell to fire an action potential. It needs some 30 millivolts of depolarization, and that 100 microvolt is going to make a small impact. However, there are also some rare synapses within the neocortical microcircuit that are much, much larger. And so we can get synapses that are 5 and even 10 millivolts in amplitude in response to a single action potential. And these synapses then could give rise to a considerable impact postsynaptically and contribute strongly to driving postsynaptic action potential firing. And so we're left in an interesting situation where there's a few very strong synapses and there are very many very weak synapses. And at this time, it's still not completely clear which synapses are most important in terms of driving neocortical microcircuit activity. Computational studies suggest that one can remove all of these small synapses and it makes very little impact upon the neocortical microcircuit activity. It may therefore be that larger amplitude synapses are the main drivers for reliable sensory processing in the neocortex, and it may be that a very sparse set of connections, which are large in amplitude, might dominate over a sea of very small connections that make almost no impact upon the larger neocortical microcircuit. So large unitary EPSPs might be of fundamental importance for reliable signaling in the brain. Further indications that reliable processing might occur through these big amplitude connections comes if we look at the variance of the PSP amplitudes. And here we compute the coefficient of variation, which is equal to the standard deviation over the mean, in terms of the trial by trial EPSP amplitudes that we measure for the same connection in response to a given action potential. And you'll remember that there's some variance. And we've already discussed that large synapses might have low variance, whereas small synapses have much more variable responses. And so that then is another indication that if you want to have reliable sensory processing, you might want to use these big connections that not only have a big impact on the postsynaptic neuron, but they're also reliable with low variance from one trial to the next. So here in this video, we've seen some important aspects about glutamate synapses. First of all, we've seen how glutamatergic synapses are essential for relaying all sorts of sensory information from the periphery to the neocortex, where it's thought that sensory percepts are generated. So glutamatergic signaling is essential for sensory perception. We've also seen that within the microcircuits of the neocortex, there are highly specific circuits that are made, where some types of neurons like to talk to other types of neurons, but might not want to receive information from other types of neurons. And so there's a great deal of cell type specificity in terms of where glutamatergic signals are made and how the sensory processing is likely to occur within the neocortex. In addition, we've seen that there are some extremely large amplitude synapses that are 1 or 2 orders of magnitude larger in amplitude compared to the large sea of small synapses that are present in the brain. And these large amplitude synapses might be particularly relevant for reliable sensory processing within cortical microcircuits. These synapses may in part be determined by genetic mechanisms. The different cell types express different genes, and so genetically one can distinguish different cell types in the neocortex, and there may well be genetic factors that skew the wiring diagram of the neocortex in one way or another. It's also very likely that experience makes a big difference to the wiring of the neocortex. We learn how to perceive the world around us, and it's likely that some of that learning as to how to interpret the sensory information occurs in the wiring diagram of the primary sensory areas. And so the distribution of large amplitude and small amplitude synaptic connections in the brain may in part relate to experience-dependent learning. And over the next video, we're going to see how synaptic plasticity through different patterns of activity can alter the strength of synaptic connections.